Thank you for joining us today. Like I said, my name is David Bourdon. I am a staff scientist at ThermoFisher Scientific, formerly the Legacy Life Technologies in Carlsbad, California. And I'm going to be talking today about some of our very unique immunoassay tools that are used in biomarker discovery and validation. You know what is really thank you. So just a very quick biography. Just want to let people know that our interest in biomarker discovery is long-standing. My background is a PhD in pharmacology, focusing on cell signaling, GPR signaling, and thrombosis. Most importantly, I've spent time leading Luminex XMAP technology assay development. And these experiences, both in multiplexing assay development and in the pharmaceutical area, looking at inflammation, have been very useful as we look at new technologies. And so I joined Life Technologies or ThermoFisher Scientific in 2009, and I've been part of a next generation immunoassay team that's very keen on developing the next generation immunoassay tools for proteomic biomarker discovery. But our current activities and work that we have performed over the last several years include the development of our Protoplex immune response assay and how we use that assay along with our cytokine panel that we have led development in for a number of years. And most importantly, these experiments or these technologies are now being used by collaborators as well, and that's how we develop new technologies, is working very closely with key opinion leaders in the areas of translational research to better identify unique biomarkers and how they can be utilized in the clinic. Okay, so very quickly, I just want to go over, many of you know Legacy Life Technologies and now ThermoFisher Scientific as a leader in next generation sequencing technology. But my area, or our area, the team I'm a part of, we look at proteomic biomarker discovery. So we're very interested in the detection of protein at a very low level, and that's very important in translational research because we understand that being able to identify proteins earlier in a disease process is key to developing treatment regimens, developing new drugs, monitoring treatment efficacy, etc. And we all realize that our products are of no use unless we can accelerate your ability to take your discoveries into publications, into the clinic, and develop new and exciting drugs. So we're, as a tool builder, and let me just say that we are not rheumatologists, we're not oncologists, but we're very proud of our unique skills in developing some of the most sensitive and most precise assay tools on the market. So we take this very seriously, and we are keen on making sure that you find our immunoassay tools to be the most useful for you. And so the key areas that we focus on are inflammation, cancer cell signaling, and autoimmunity. And so I'll touch a little bit on these three areas of emphasis today. And I'll finally wrap up with something that makes our offering very unique. And this is something that as an R&D scientist, I'm very proud to represent our entire team in the accomplishments we've made to demonstrate unique utility using our multiplex immunoassay technology. And in other words, what I'm going to talk about is how we use Luminex XMAP multiplexing, not only for detecting cytokines, but also for looking at autoantibodies and using these data sets together to give very synergistic outcomes. In other words, we're able to identify unique biomarker panels in certain disease areas that we think you would find very beneficial. So everybody on this call, on this webinar, probably is very familiar with the means of inflammation and how inflammation can affect our bodies in disease. And so I don't need to go into great detail on this. We know that chronic inflammation is a classic response between pro and anti-inflammatory extracellular mediators. Here's just a short list of these mediators. This topic of the importance of inflammation has been discussed in popular literature and, of course, all the key medical journals. And so inflammation and how it can relate to cancer, of course, is very important to nearly every drug, pharmaceutical company, medical research institution, et cetera. And what we're trying to do is make the best kits that will allow you to accelerate your research. And so, I think we've all experienced various forms of inflammation, just everything from a bee sting to a puncture, let's say a sliver, et cetera. But what goes on under the plasma membrane surface, the intracellular signaling that goes on, is quite complicated and quite complex. And being able to elucidate these pathways is sort of one of the hallmarks of multiplexing immunoassay technology, being able to understand how a extracellular signal passes through the membrane through, of course, receptor molecules, and how those signaling pathways can lead to changes in expression of protein through regulation of transcription factors, etc. And so there's over the years been a growing collection of peer-reviewed journals that highlight the roles of chronic inflammation and how that can lead to various forms of cancer. And I'm not going to go into great detail about some of these seminal papers, but we know that there's important correlation between chronic inflammation and cancer. So the reason that is important to mention is that many of our immunoassay tools are measuring, for example, extracellular cytokines, and people may use those cytokines in classic inflammatory disease research. But many of those same markers, many of those same cytokines are being used by key labs to examine the etiology. And so this is a paper, a recent paper, last year, 2013, from MD Anderson, a very nice review article highlighting some of the STAT pathways that are involved, phospho-STAT pathways that are involved in regulation of gene expression, pro-inflammatory gene expression. And so the reason I mention this is that in this article, they give a very nice overview of two different cohort studies in which a number of different classic inflammatory mediators have been linked to chronic lymphocytic leukemia. And that's a form of cancer that I'll talk a little bit later about because our 30-plex cytokine panels have been used by a number of investigators to look at various forms of cancer, including chronic lymphocytic leukemia, or CLL. And so this is just a recreation or a snapshot of the figure, the Hanahan-Weinberg paper from 2001, and of course they came out with another paper 10 years later, and they have been thought of as some of the leaders in the cancer field for a number of years, and they have categorized a number of different events that are classic to the study of cancer. And the reason I mention this is over the 10 years, as Hanahan and Weinberg outlined, many great discoveries have been made. But at the same time, there are other things that still remain elusive. And what we are interested in as a corporation, as a tool provider, we're interested in providing cutting-edge technologies and assays to measure these particular markers that can be involved in a number of these different steps, including tumor-promoting inflammation, angiogenesis, etc. And so what I've done here is I've just highlighted some of our kits and some of the markers that are involved in this hormone. So as far as technologies and tools, so I'm doing a little bit of a switch here. It's very important to look at a number of different markers, but the methods by which we look at those markers, the tools that we utilize, can really depend upon specific needs. So you, as a scientist, can choose from a number of different immunoassays to consider. So some may consider classic Western blotting because they do need that size confirmation of being able to examine proteins that are immobilized on a nitrocellular membrane, for example. And the workflow of Western blotting is good enough, and it's actually very conducive to the workflow necessary. In other cases, a researcher may need a much more sensitive approach, something that's more quantitative, like the class ELISA. And again, other people may be looking at multiple markers. For example, translational researchers looking for new biomarkers may actually be panning for multiple markers, looking for one or two markers that is truly indicative of disease progression. So whether you need sensitivity, richness of content, throughput, or workflow, we offer quite a few different choices to choose from, or choices in immunoassay tools. And so all of those tools involve affinity reagents, and these affinity reagents can target a number of different molecules, cytokines, chemokines, receptors, etc., from a number of species and a number of sample types. And the reason I bring this up is because we all know that being able to detect a protein at very low levels in serum or a cell lysate can be very different than plasma with EDTA, citrate, etc. So we have a lot of experience being able to measure proteins at very low concentrations in a number of different sample types, and being able to understand the spike in recovery and the performance of that assay in different sample types is critically important. And we really pride ourselves on providing all of this information to the customer. And so these parameters by which we measure our immunoassays are listed here on this next slide, so numbers 1 through 10. In the interest of time, I will not be going through all of this during one example, but I do want to say that our hope here is that this presentation today is just the beginning of longer discussions and a greater relationship with our end user base, that we really do want to help. And so we really would enjoy talking to you more. We have a sales force around the world that's able to get you in touch with researchers like myself and really help and narrow down what assay is best for you. And so these are all the specifications that we must perform, and we feel that we, excuse me. Okay. I'm being instructed to speak a little bit closer and louder. Sorry if, how's that? Okay. So anyway, I'll cut through the chase real quick here. So there are a number of different specifications that we try to, we actually do meet and exceed. And so one of them I'm going to talk about today, I have all of these slides. We don't have time to go through all of them. However, one of them is critically important to talk about in this forum, is our calibrated standards. And so it's very important to note that all of our protein standards are calibrated against NIBSC. And so one may ask, well, what is NIBSC and what's the point of calibration? Well, the point is that if you buy an assay from any vendor and provider, it's important to be able to assure yourself that when you perform that assay, let's say you purchase an assay in June of 2014, and you get some great results. You want to make sure that when you use those same assays, you buy a new kit 3, 6, 12 months from now, that you can ensure that the performance will be the same and that you can compare those different data sets. And the only way you can do that is with calibrated standards, because we do post-lyophilization calibration, so the bioavailable amount of the protein in the standard curves is calibrated against the same gold standard during the manufacturing process over years to come. So NIBSC is an organization in the United Kingdom, and what they do is they sell calibrated standards. And so this standard, for example, let's say TNF-alpha. They sell TNF-alpha, or they provide TNF-alpha to a number of reference laboratories. And each of those reference laboratories is given, let's say, a 10 nanogram per mil standard protein mixture. And they ask all of these reference labs to measure the protein levels and report back to NIDFC what they believe that number is. And so some labs may come back and say, okay, that I believe that is not 10, but 9.2. Some may come back and say, you know, that's really more like 10.5. Well, what NIDFC does is take the average of all those results, and then that is their gold standard calibrator that then they sell to assay providers like ourselves. So when we manufacture, we express and purify our TNF-alpha, for example, we then calibrate our protein against NIDFC. So we're using that same calibration as a gold standard constant throughout the process, and year after year, every kit is calibrated against the same. So the reason that's important is when people are doing longitudinal studies, they expect our assays to perform the same year after year after year. And to us, that's just a given. It's obvious that we must ensure that same reproducibility and precision year after year after year. And so I've had some nice discussions with various core facility managers in the United States, for example, and they're very pleased to hear that we calibrate our standards so that them, as a core facility, can ensure the quality and that their customer base, that is the scientists at their institution, are getting the highest quality results. Excuse me. So that's just one of the many metrics that our assays must meet. Okay, so I'm going to kind of take a break here for a second and grab a drink of water. And what I want to do is switch gears a little bit. And so just in the interest of time, I'm not going to go through all those other assay metrics, but it's important to note that you must ensure you have a very high quality immunoassay affinity reagent, and that the protein standards are calibrated. Now you're ready to build your immunoassay tool of choice. And so, of course, over the years, we've created and sold a wide range of ELISA kits, and those same antibody pairs or antibodies can be used for Western blot analysis. But today, the translational researchers are really interested in getting as much information out of each sample as possible. And that's where multiplexing is critical. And so, for example here, if you're trying to measure a collection of 4 different cytokines, let's say these 4 I list here on this slide, the classic techniques would include just using sandwich-based ELISAs in a classic flat bottom microtiter plate. But of course, because of the detection scheme, one must spatially separate each of the capture reagents in different wells. There's no way to distinguish one detection signal from another in the classic sandwich ELISA. And this becomes even more of a challenge if one wants to measure even more analytes. And particularly in this case, we know that many disease researchers want as much information out of their precious samples as possible. Many translational researchers are doing clinical trial studies, and the main limitation is sample amount, and also time, money, etc. So they want to get as much information out of a single sample at a single time point as possible. And that's where we have worked on making that even more effective. And the technology I'm going to talk to you about today is one of many technologies that we have, that we've been working on. And there it goes. So if you think about a classic sandwich ELISA, now looking at a 16-plex panel, one has to run, in this example, 16 different ELISA plates, which is very cumbersome and expensive, etc. And so one does not want to do that. In fact, most translational researchers today want to be able to condense all of that data into a single well. Well, in order to do that, we have licensed Luminex technology from Luminex Corporation. So Luminex is a company in Austin, Texas, in the United States. And for about the past decade, the intellectual property and their specialty is around the ability to very reproducibly manufacture beads which are about 6 microns in diameter. But more importantly, they've worked out a methodology to dye the beads And I'm only showing one example. This is their classic 10 by 10. But they also have a technology where they added a third dot. So it's an X, Y, X, Y, and then Z, a third dimension, which is a third dot. But in this example, it's basically a concentration gradient of an infrared dye from low to high, and a concentration gradient of red dye from low to high. What you see here is this is low, low, high, high of the two different dyes. And what that allows you to do, so once the beads are dyed and then rinsed, of course you can do anything you want to these beads because the instrumentation will use lasers to excite the fluorophores, measure the emission spectra, and each one of these colors on the beads is sort of like a fingerprint. It's a unique identifier that identifies this bead type from this bead type, for example. And you may ask, well, why would you want to do that? So the key here is that you can now use this bead type. For example, this is number 19 here, 1 through 10, 19. You can use that to conjugate anti-IL-2 capture antibody. And this bead type here, you can use to conjugate anti-IL-4 antibody. There's about 3 million binding sites on the surface of the bead. And you can see here that example of a 4-plex cytokine panel, you actually create 4 different sandwich ELISA scenarios, but all on a bead. Why would you want to do that? Well, you can actually, because each bead region or bead type here, the color, can be identified by the spectral emission of the dye. So you can actually mix all these beads together and perform a classic sandwich ELISA, but on a bead in suspension in a single well or a single tube. And what happens is the software, the instrument, the data analysis software actually deconvolutes the data after the fact. So you're still measuring all of these binding events. And of course, the first fluorophore here is going to help identify the bead type or the bead region. The second fluorophore is purely for detection. And so this is the second in the antibody pair. This antibody is directly conjugated to cyclobutadiene. So this is a red channel, approximately 635, and this is the green channel, approximately 532. And how that works is after you perform your classic immunoassay with sample addition, incubation, capture, wash, detection scheme with a detection antibody, wash, you run the samples of a bead mixture in an analyzer. And in this case, the Luminex 100-200 instrument is actually just a classic flow cytometer that allows you to excite the bead region fluorophore and the detection fluorophore to both identify the bead region and the amount of binding that's occurred on that bead. So, of course, the amount of detection signal is proportional to the amount of analyte that's been captured from the sample. And so each one of these beads comes in a flow cell, and each bead is read one at a time. This happens very, very quickly. And so our assays will read 100 beads, and so each event, each counting event, will be 100 individual beads. Again, there's about 3 million types per bead. So you're measuring many, many binding events. The other platform is the MagPix, in which it's purely for magnetic beads, and the beads in the well are then deposited onto a flow cell here, and with the magnet in the instrument, all the beads are pulled back to a monolayer. And between the LED green channel and red channel, you're achieving the same goal as in the 100-200 instrument, but it's basically a micro array. So there's 2 different ways you can read out the bead technology. The key here is that the value of multiplexing is getting very rich data sets with a few number of samples. So this is just a very simple example of 7 serum samples that are being run from our cytokine panel. And you can see here, classically, each one of these bars would be indicative of a single ELISA well. But now, they're able to get 25 different data points in that same single well. Just as importantly, for cost benefit, you can imagine as you increase the number of analytes per well, the overall cost of the assay and the per analyte cost goes down considerably. So not only are you benefiting in reduced sample usage, but there's also, it's a more efficient process for how you spend your research dollars. So the first question that many people ask is, well, okay, this sounds great, but you know what? It sounds too good to be true, and there's probably a lot of cross-reactivity with antibodies. And of course, that's one of the greatest challenges of any of these kits, is being able to find robust antibody pairs that do not cross-react with each other. And this is just a great example of our 9-plex cytokine panel. We go all the way to the 30-plex, and we're constantly building higher plex panels. But this is our 9-plex cytokine panel, where we're demonstrating little to no crosstalk between the bead regions and the cytokines. So these are on the top line here are each of the beads conjugated with a capture antibody for each of the cytokines. So anti-IL-10, anti-IL-13, etc. And each of those bead types is then incubated with either IL-10, 13, etc., each of the 9 cytokines. And what you're seeing here is very nice signal noise. So this would be the background signal. But this is a very nice specific signal, very high signal noise. Another thing people get concerned about is, okay, when I'm moving on to a new technology, you know, what's the value in being able to correlate to, you know, if somebody's been measuring samples for years using some of our ELISA kits, what's the conversion barrier and things like that. And actually, the great news is because we're using the same high-quality antibodies from our ELISA pairs in our Luminex platform, the concordance is very, very good. And so it just makes sense that when you start with high-quality affinity reagents, you're going to have a high-quality assay. And so this is just a quick snapshot of our technical data sheets. We provide this for all the kits that in. I wanted to point out here, I mentioned earlier NIBSC calibration. I wanted to point out here are the NIBSC calibration values for this kit. In one case, for IP10, when we generated this production lot, we did not have a calibration number for that. Of course, that can change over time. But the point is that we provide all of this information about recovery, sensitivity, NIBSC calibration, assay variation, all of that to you. So you have all the information that you need. And so up to this point, let me just say that, sorry if the talk's been a little bit of a sales pitch here, but at the same time, I think we're very proud of the way our assays perform. And when I talk about science, so at this point, I'd like to sort of switch gears and talk much more about what we are concerned about at the lab bench and what we're constantly working on to make even better and make more powerful. Is what I wanted to do is sort of set the stage to say, really, when you're comparing technologies, and frankly, I've been in your shoes, I've been on that side of the bench, and I know that you're confronted with many different choices, a number of Luminex partners, a number of immunoassay tool providers, and some of it can come down to the price, et cetera, of course, but more importantly, it's all about quality. And we know that many times you can only perform an assay once, you have very precious samples, and you don't ever want to repeat an assay. And so when you have high quality affinity reagents, the assays are well built, and the quality is maintained over time, we feel that you're getting the best value. And so that was just my sort of internal company pride sales pitch. But now that you know that we've got a very good assay to work from, I wanted to talk a little bit more about how our kits are being used. And this is very exciting for me as a scientist to see our kits, because frankly, some of the kits and some of the technologies that we're working on right now for next generation tools, our goal, my teammates, myself included, our ultimate goal is to see those kits and those tools being utilized by people like yourself daily. And when those discoveries are made using our kits, our tools, and they are presented in top-notch, peer-reviewed journal articles, and that work can then lead to new therapy development, of course, and then bettering human health. It comes down to one of the core values of our corporation, and as a bench scientist, we can't be more proud to see our products used in a very positive way. And so that being said, I just wanted to show a couple really neat examples of how our high-plex cytokine panels are being used by our user base. And so this is just a list of a very short list of the articles that were published in 2012 by key researchers using our fix. But let me go into a little more detail on a couple. So this is a paper that came out a couple of years ago in which our users, the customers, actually wanted to do a retrospective study. For those in the biosimilar space, many of you are familiar with the classic PGN-1412 tragedy. About a decade ago, there was a drug company, a biosimilar company, that was looking to develop a monoclonal antibody against CD28. They considered this to be a T-cell specific marker, and it lacked co-stimulatory signal recruitment. So they were hoping to use a humanized monoclonal antibody against CD28 to help target for autoimmunity and chronic lymphocytic leukemia, the disease I had mentioned earlier that is closely related to inflammation. TGN-1412 was designed to bind and stimulate anti-inflammatory cytokines and expand Tregs. Unfortunately, there were a number of adverse events in this clinical trial, and there were fatal consequences. And so that was about a decade ago. And more recently, this group from Glasgow did a very nice retrospective study using our 30-plex cytokine panel to help better understand what exactly happened and why did these adverse events actually occur. And so what they did is they performed a number of studies, and I'm just showing one of many great pieces of data published in this paper. But they had they had monocyte cultures that were being treated with the TGN-1412 drug, and then they used our 30-plex cytokine panel to measure levels of these cytokines, pro-inflammatory, regulatory, chemokines, etc. Long story short, they found quite a large number of these inflammatory cytokines and other mediators that were elevated after the administration of TGN-1412. So what actually happened was, presumably in the patient population, they were administered the biotherapeutic antibody, TGN-1412, which resulted in monocyte recruitment to the vascular endothelium, which then led to a release of a number of cytokines, activation of natural killer cells, and really led to what they call a cytokine storm. And so, again, I don't mean to bring up this. This is a little bit of it's a very serious outcome initially a decade ago, but it was very interesting to see how some of our users have used our kits to add to the knowledge of this drug. And I'm sure I've talked to many in the biosimilar space. This TGN-1412 tragedy is well known, and many. Oh, okay, thank you very much. Many changes in how biotherapeutics are made have resulted. Secondly, this is another quick paper that people were looking at our 25-plex panel to study chronic lymphocytic leukemia. And so, long story short here, they're looking at 84 CLL patients and 49 age-matched controls. They use time to first treatment as a key indicator of disease severity. They ran our 25-plex cytokine panel against all of these serum samples, and they identified certain clusters of cytokines that were observed to be upregulated in the more severe case of CLL. So cluster 1 is present in higher concentrations in a more severe form. Cluster 1. Clusters 2 and 3 are actually elevated in the less severe form of the CLL. So again, it's just a great example of how our kits are being used to better understand inflammation and chronic lymphocytic leukemia. Okay, so besides the extracellular signaling molecules, there's a lot to be said for studying what's on the other side of the plasma membrane. So all of the kinases, JAK-STATs, AKT, for example. I won't go into great detail, but we do have some great tips to look at these classic signaling molecules. And for example, this is our phospho-AKT 7-plex. So we have a total and a phospho-AKT 7-plex. In the interest of time, I'll sort of speed through this, but again, all of the slides are available on a website that I'll show you address at the end of this presentation. But the power of our multiplexing, even at the intracellular level, can be demonstrated here. We all know that AKT is a very important signaling molecule. This is the data to go with. We also have kits to examine ERK1 and 2 and P38, as well as JAK-STAT pathways. And so it's important to note that just taking a step back from our bead-based assays, you know, because we're using the same high-quality affinity reagents, not only in our ELISAs and our Luminex assays, but those same antibodies can be used in Western, flow-in imaging, et cetera, because we realize that when you're going to publish a paper, you need to show several techniques or several methodologies to demonstrate a phenomenon. The peer-reviewed journal committees require it, and so we want to be able to provide the most comprehensive tool set for you. And finally, I just put together a quick demonstration here to show the number of signaling molecules that can be measured using either our antibodies, ELISAs, or bead-based Luminex assays. And that's a very good point that we actually just launched a new human adhesion 6-plex panel. The SKU can be, you can track this down on our website, but it's very important for inflammatory disease researchers to better understand these increase or decrease in the presence of these adhesion molecules, not only for inflammation, but also for metastasis in cancer research. And finally, our online tools provide even more access to custom assays. So you can even custom tailor your own assay. Okay, so finally, the last bit I'm very excited to talk about and help represent my entire team. We've really done some interesting work to better understand how we can put together different types of signaling molecules and look at them. For example, how do we bring more clarity out of this chaos? So this cartoon here just kind of shows the intricacies of the inflammatory of the immune system. And what I've been talking about throughout this webinar is really everything intracellular and extracellular, with exception to plasma cells and IgGs. And that's a very important aspect of the inflammatory response as well. It's something that we do very uniquely, that we're measuring both antigens and autoantibodies. And the reason autoantibodies are so important, this is a beautiful review article from Karen Anderson in 2012, where she highlights the importance of autoantibodies in the study of cancer. And so for anyone who's unfamiliar with autoantibodies, they're an IgG or an antibody molecule that is being released by our immune system in response to the increased presence of a tumor protein, for example. And here's a list of a number of different autoantigens that are associated with changes in disease progression. And so we've developed a number of tools to look at the presence of autoantibodies, and our protein microarray, protoarray, is one of our leading technologies. It's a 1x3 glass slide that contains about 9,400 human proteins spotted in duplicate. And so this microarray allows us to measure human antibodies that will bind to those proteins. And so what I'm showing right here in this slide is that, for example, everyone has been exposed to various forms of the influenza antigen. However, people that are suffering from a disease such as lupus may have a unique immune response to a protein called Rho52. And so this is a great example of how protein microarrays, protoarray, has been used to distinguish or stratify patient populations, for example, healthy versus a certain disease. But what's been missing over the years, and so protoarray has been around for quite some time, but what's been missing, and by the way, we just came out with a new version late last year, version 5.1, but what was missing up until about a year ago was the ability to move on from discovery to verification and ultimately validation. Because if you think about it, the classic biomarker discovery to validation workflow starts out with very few number of samples, let's say on the order of 20 to 100 samples, split between healthy human control and disease samples. but you're using a very high content pool. For example, in the case of ProtoArray, which has over 9,000 potential biomarker targets. Once the hits are identified in technology like ProtoArray, one must go on to verify, ideally in a different patient cohort, identify and verify the presence of those same autoantibodies in an independent sample, with the ultimate goal of finding just a few biomarkers that they can run in a very large number of samples. So right on this end of the spectrum is more like, let's say, 10 to 100 samples. But out here, you want to get out into the thousands of samples. What's been missing is something in the middle to look for verification. And what we developed several years ago is Protoplex. So Protoplex is basically a Luminex-based platform to present the same autoantigen, the same proteins that are on ProtoArray, but in a bead-based platform in which one can multiplex. The point here is after you've identified, let's say, 20 to 40 markers on ProtoArray, you would then take those same proteins, attach them to a Luminex bead, incubate them with other serum samples to verify that those same autoantibodies are present. So this is the human autoantibody. You wash the beads with buffer, and then you come in with an anti-human IgG that has, again, cyclourethrin, which is the classic out for Luminex technology. So we developed Protoplex to be a very flexible yet robust verification and validation system. So it's designed to work on any protein content, any protein, any source. Of course, we have our Ultimate Orth collection, which is approaching over 20,000. And so the proteins that are used in ProtoArray can then be attached to a Luminex bead, either directly conjugated or through an epitope tag. And then, because we're not dependent upon the same antibody pair phenomenon that you would see in a normal cytokine capture assay, we can actually utilize the full 100-plex that Luminex has as their standard technology. And just as importantly, we've developed a number of key data analysis tools, and we have an amazing bioinformatics team that's developed some nice algorithms for looking at very low-level detection, not only in the cytokine realm, but also in the autoantibody realm. So we're able to do instant group-wise comparison. And this is just a very quick snapshot of something that we're looking to develop and launch down the road. We're going to have a data analysis tool set that goes with both our cytokine panels as well as our autoantibody panels. So finally, one thing that we wanted to do in-house within our corporation is to demonstrate the utility of not only looking at autoantibodies, like it's been described previously, the value of autoantibodies in there, but especially in the case of lupus. Lupus has been very elusive. To this day, there is no one telltale blood test or diagnostic that's used to characterize somebody with lupus. So what we were hoping to demonstrate is that by looking at not only autoantibodies, but cytokine panels, we can maybe bring some clarity to that chaos of identifying biomarkers in lupus. And so what we did is we acquired a number of serum samples from bioreclamation, and we used, so we had age-matched healthy controls and those that have been well curated and diagnosed with lupus. And those serum samples were run through our 30-plex cytokine panel as well as our 19-plex autoimmune panel. And the data that we observed was very interesting. So we found about 5 cytokines and 9 autoantibodies that are significantly elevated in the lupus population with respect to the healthy human control population. But what was first exciting, but then indicated to us the challenge that exists today, is that in order to identify a panel of cytokines that is very strongly predictive of the disease, we had to find a 5-plex cytokine panel. That same phenomenon of a large panel to get high predictive value was even more exaggerated in the autoantibody panel. However, if you take those same serum profiles, those same serum signatures, and now not look at them separately, but together, in other words, at 49 serum profiles together instead of 30 and 19 separately. You can see here that we actually are able to identify a unique two-marker panel, one cytokine and one autoantibody that are still 98% predictive of disease. We were very excited about that. We've actually filed a patent on this approach. We're not rheumatologists again, so we were more interested in filing on the methodology and the means by which we came to this result. And so what we're doing now is I'm working with two researchers in cancer, translational cancer research, to now use the same approach to look for unique biomarkers in those diseases. And we'll be using, instead of an autoimmune panel, we're using a cancer panel. And again, cancer panel along with our 30-plex site panel. Okay, so as far as a unique offering, I think that's what we're really proud of, is that we're taking the data sets and using them in a very synergistic manner. This is something that you won't see from other companies, in fact, other Luminex partners. This is something that we really feel like we're on the cutting edge, and we learned a lot about what translational researchers absolutely must have in next-gen tools and current tools that we offer. And so this is just a nice high-level cartoon of our antigen and autoantibody detection schemes. And so today I talked about a number of different technologies, everything, of course, from our proteins all the way up through our protein microarray. But I'm really hoping that this presentation just gave you a good introduction to our technologies, but it also is just the beginning of a great dialogue that we can have with these individual researchers. And finally, our new Immunoassay brochure is available on our website. And here's the, so we have a new Science of Scientists portal that we've created. And right now, it's just featuring the work that I've discussed today. However, in the months to come, you'll see other areas presented here as well. But at this website, you will be able to download a copy of the slides that were presented today, and actually a little bit more about what we're doing. And so I really appreciate your attention. And if you take time for questions, I'd be happy to discuss them or clarify anything else that you've heard today. Thank you very much.